So it's my great pleasure today to introduce Dr. Latania Logan from Emory University. So hopefully I get all of this right. Dr. Logan performed her undergraduate studies at University of Michigan, completed her MD from Wayne State University, and her Master's of Science in Public Health from the University of Illinois at Chicago. After completing a residency in pediatrics and a fellowship in pediatric infectious diseases, she then joined the faculty at Rush University Medical Center in Chicago, where she served as a clinician and led research on the clinical and molecular epidemiology of multi-drug resistant bacterial infections. Then in 2022, she joined the faculty in the Department of Pediatrics at Emory, where she has been continuing her work on antibiotic resistance. So I invited Dr. Logan here to fill out our webinar because I thought she could provide a really interesting additional perspective as both a clinician who is doing and also a researcher who is doing exciting translational research looking at community reservoirs and environmental transmission of drug-resistant bacteria. So with that, I'll let her take over, and I look forward to her presentation.
Thanks so much, Liz. I really appreciate the introduction. Thank you guys for having me. And I just want to note, they know, but I'm on clinical service at the hospital. So if I have to take my phone and give it to somebody, just know it's not because I'm not paying attention. I just have to make sure some questions get answered. So I'm going to share my screen now. Okay, can you see me? Over the screen? So there?
Yes, you're good to go.
Okay. All right, wonderful. Okay, thanks again, everyone. So my lecture is about MDR Enterobacterales in the children and community, and I'll talk a little bit about that. So I don't have any disclosures. So the objectives today for me are, I want us to define the problem in terms of MDR Enterobacterales. We need to understand the organism. We'll talk about the role of discovery, and then we'll go into how we find the sources for infection, and finally about understanding what are the health implications of these things for humans, and we'll talk about the whole idea of One Health here. So let's start with defining the problem. So you've seen this before. Dr. Atkinson talked about this as well. So, we know that this is a problem with antibiotic resistance, and I won't go into these details here, but you will. I don't know if you know, the amount of costs that are actually associated with these drug-resistant infections. And so of that list of the CDC AR threats report, where there are 18 organisms, just 6 of these infections, $5 billion annually. So it's a problem for all of us. So the good news is that since the CDC put out that threat report between 2013 and 2019, we found that prevention really works. There was a lot of reduction that occurred, especially in healthcare settings with interventions from CDC, the Departments of Public Health, and the epicenters. However, the bad news was that what we also found was that there were other things that were increasing during this time. And in particular, there were a lot of these community-associated organisms that were increasing, and that's where we're going to focus our attention today. So we talked about this also a couple weeks ago, but this is an important thing. The bugs are getting smart on us. So the antibiotic resistance, back when we started having antibiotics, it took 20 years on average for the bacteria to get smart on the antibiotics. But up to when you talk about 2020, less than a year it takes before we start to see resistance in some of the newer agents. So I won't go into this, but you can see these are the 18 threats, and we are going to actually focus on these, which are the beta-lactamase producing Enterobacterales. So the beta-lactamases, these are drugs that are of about basically 5 classes that we consider plus minus some of the newer ones, but these are basically beta-lactamases that are produced by the bacteria, are enzymes that are able to break down beta-lactam antibiotics, and they hydrolyze the beta-lactam ring, and that renders the drugs ineffective. You can actually find this in both gram-positive and gram-negative organisms. But I'm not sure you know that there are more than 3,000 unique beta-lactamases that have been identified, so that's definitely keeping me in business. So the ones that are in the gram-negatives that are important are called ESBL-producing Enterobacterales, and these are the ones that are in the threat report and the ones that we focus our attention on. And what's important about these is that these are organisms that have been increasing over time, really since the early 2000s. But the difference is they have gone from being really a problem like MRSA, where it was just part of the healthcare associated infection world, and then became community acquired MRSA. This same sort of thing happened a little bit later with these ESBL producing Enterobacterales. And these are very drug resistant. They are resistant to multiple classes of the beta lactams. And so the drugs that are used to treat them are less. And there are multiple classes and types of these that are multi-drug resistant. So what's interesting is if you look at that graph on the right, you can see that the community-associated infections are really a predominant part of where we are seeing these infections increase within people since 2019 and the threat report. And so the question becomes, why? Where are these drug-resistant bacteria coming from? And that's what my research focuses on. So the one thing that we started out was trying to understand if these were a problem of infections in children, because there at the time when we started doing these studies back in about 2013, was that there was just no data on what was going on in children. So what we did is work with the Centers for Disease Dynamics, Economics, and Policy, what is now the One Health Trust, is out of Washington, D.C., looking at trends of resistance in children across the United States. And basically we used SIR, or antibiotic susceptibility data. And this was a very robust lab system of 300 labs across the nation. And so what we saw was when you looked at third generation cephalosporin resistance, which is a phenotypic proxy for ESBL infection. So some of these large, higher class broad spectrum drugs of the cephalosporin family. And then you can kind of get an idea, but not all of them are ESBL producing, which is why these are a little bit different. But looking at the colors, what you can see is basically, if you look between the time period of 1999 and 2011, you can see basically the resistance marching across the nation in the children. So all the regions were seeing increases in drug resistance, and we found that happening in all demographics and age groups, including outpatients, where the largest increases were occurring in the 1 to 5 year olds. So the youngest of the children. And when you looked at where the children were located in our study of children out of Chicago, and looking at these drug-resistant infections, what we found was similarly, these children were very young in age. The median age was 4.2. And we saw that there were basically 27% of them were infants. 60% were female, and you can see that when they presented, almost half of them also presented in the outpatient setting. So the types of infections caused by Enterobacterales are important because we know that if you look at the graph to the left, that it can cause any type of infection. But most of the infections we're going to see are gastrointestinal and urinary tract infections. One important thing is the difference between what is associated with healthcare infections and what is associated with community-based infections. And so if you look at healthcare-associated infections, length of stay is important, invasive devices for getting those types of infections. Also, neurologic or urologic comorbidities are also associated with these types of infections, or any chronic illness, really. And then antibiotic exposure is important. Next, please, Lydia. Yeah, thank you. So, so this is what I found and what is in the literature also to be based on community-associated infections, which is different. And this is where my research focuses, and we'll talk a lot about these things. But maternal child transmission, household transmission, environmental exposures are very important. And these we will all talk about. International travel is a big reason that people come back with these infections from endemic regions. And again, antibiotic exposure is very important. Next slide, please. Okay, so now we're going to move on to understand the organism and mechanisms of virulence and resistance. Okay, next, please. So the beta-lactamases, these are the major driving force of transmissible resistance in gram-negative bacteria, and there are basically structural and functional classifications for grouping. And so there are also differences in the types of mobile genetic elements that these organisms harbor. So while we talked about this ESBL pandemic, that actually is most commonly occurring in E. coli, but there's also this problem with carbapenem resistance, which are due to these carbapenemases, and that pandemic is actually occurring in Klebsiella pneumoniae. And so this is very complex, and I will try to discuss this a little bit more in a couple slides, but the biggest contributor to the global expansion of these multi-drug resistant Enterobacteriaceae or Enterobacterales are clonal strains. However, there can be promiscuous plasmids in combination with other genetic elements that are propagating the drug resistance. And this is actually known as horizontal gene transfer. And so this is actually going to be a big component of the problem in children. Next slide, please. Okay, so in our study in Chicago, we looked at these beta-lactamase genes in isolates where we only had phenotypic resistance profiles from the children. And we basically got this from the 5 microlabs, and we looked at genetic markers of resistance. So we did PCR-based tests and DNA microarray to understand the beta-lactamase gene types that were responsible for these infections in children. And so what we found was the majority were part of CTXM group 1. Now that is part of the CTXM producing ESBL producing Enterobacterales, and those are the most common clonal strains that are circulating within the US and actually within the world. However, that CTXM15, while it's within group 1, this clonal strain, the ST131 strains that tend to carry that specific allele, they're not the only ones that do this. So there are lots of things that the CTXM E. coli actually carries. So group 1 is not just CTXM15, and this is important because there are multiple strains that are circulating, and we really don't get that information unless you have a lab that can actually look for CTXM, and then they can't necessarily tell you the allele, but they can tell you that it's part of that group. But what I want you to see here is that what we found within the kids is that there were multiple types of beta-lactamases that were causing these infections in children. And this is important because it's not a one-size-fits-all. This means that there are different routes of transmission likely occurring, and the children are getting these from different exposures. So we need to understand more about where this is coming from. Next slide, please. Okay, so this is are a couple graphs that I'll take a little bit of time to just try to walk you through. But when you think about the most common causes of infections due to ESBL producing organisms, those are going to be your urinary tract infections. And in general, urinary tract infections are most commonly caused by E. coli. So this is not a surprise to see that the most common organism that we found in children is E. coli. Because that's what would be found in all of us. However, you can see that there were many different types of these Enterobacterales, and you might hear me toggle between Enterobacteriaceae and Enterobacterales, and that's just the difference between going from class and order to family, and it's just the nomenclature, and it's changed a little bit over time, so I apologize if I toggle the names, but I'm talking about basically the same thing. But you can see that there are several different types of organisms that were associated with these very drug-resistant genes. And then on the right, just what I want you to notice is all the colors. And what the colors are showing that within all these different types of organisms, we were finding all these different types of genes, meaning there wasn't a one-size-fits-all. This is not just one clonal strain of ST131 E. coli circulating. These are beta-lactamase genes and mobile genetic elements that are transferring in between different genera. So there's a lot of horizontal gene transfer that is occurring amongst these Enterobacterales, and that means this is what is occurring among the children. So this is a very complex issue. Next slide, please. So one of the other things I was really interested in is in the fluoroquinolones, and that's because 50% of the isolates resistant to fluoroquinolones that had these ESBL, were part of these, cephalosporin resistant isolates. And so we were curious because fluoroquinolones are not a commonly used antibiotic in children, whereas in adults it is commonly used. And so the mechanisms of resistance tend to be chromosomal because of the bacteria just getting smart after a long-term use of these drugs in a population. So what I wanted to understand was why we're seeing these and what are the mechanisms, because there are chromosomal fluoroquinolone resistance mechanisms, but there are also ones that are on plasmids. And what we found is that there were multiple different plasmid-based mechanisms of resistance from fluoroquinolone resistance within these pediatric isolates. And so we really wanted to understand why. I will say that the AAC6 one is more commonly associated with that ST131 CTXM producing strain. So that isn't as surprising, but it is really surprising to see how much of this resistance is circulating among children and these different genes, which we'll talk about a little bit later. Next slide, please. And not to spend a lot of time on this, but just to show you how complex when I talk about horizontal gene transfer, because this is like the movement of plasmids or transposons, different insertion sequences. It's a lot of movement of mobile genetic elements that are associated with the ones that harbor these antibiotic resistance genes. And what you can see here is this is just one kid, and I just wanted to show you this one because I thought it was very interesting. So this is actually a Klebsiella pneumoniae strain. But what is really important about this is when you look at it and you do whole genome sequencing, you can see that the biggest plasmid in here is actually an E. coli plasmid. And there were multiple plasmids from other types of Enterobacterales in just within this one strain. So within this child, there is actually a lot of movement of genes and plasmids. So this is just showing that this can actually change our antibiotic resistant bacteria can affect our profile of normal non-drug resistant organisms that would just normally be colonizing and flora that shouldn't have these genes, but they can get it transferred to them as well. Okay, next slide. So our commensal flora are being affected. So the other thing that I was interested in is because I found those fluoroquinolone resistance genes, and one thing that is interesting about plasmid-mediated fluoroquinolone resistance is that it tends to be low-level resistance, so meaning the expression of resistance, like when you get your, someone says you have an E. coli infection and they tell you what antibiotics it's susceptible to. Usually what would happen if you just had one of these mechanisms of resistance, you might just see it actually be sensitive to the fluoroquinolone. So you would not know that these genes are there. It actually sometimes takes the couple of the plasmid-mediated mechanisms, or you have to have chromosomal resistance as well to be resistant. So what I did is I was curious to know if I looked in strains where it was fluoroquinolone sensitive, would I find within the children's strains fluoroquinolone resistant plasmids? And I did. And there was about a quarter of the strains, or 27% of those strains, that had these silent fluoroquinolone plasmid-mediated mechanisms of resistance. So they are transmissible, which is what is important here. So you can see that they are in different types of organisms, which is what I wanted to show you here, different types of strains types. And what was interesting here is that you can click, okay, is that when you look at the whole genome sequencing, we found more than we were looking for. Actually, there was a plasmid in here for MCR9, which is actually a mobile colistin resistance gene. And that was first discovered in 2015 in China. But people have gone back and looked and found that this was actually probably there before that time. It definitely was. And it was really associated with pigs and antibiotics related to pigs and their feed. And so this is a transmissible mechanism, and this was the first time that we had seen transmissible colistin resistance, or one that was moving on one of these mobile genetic elements. So MCR9 is one of the later ones that were discovered. But what's interesting about this is that we found MCR9 in strains among children before the first recognition of this particular drug resistance gene. So this was circulating within children long before before it was recognized in others. So this is important because children may be silently disseminating these genes and these organisms because they're less likely to get sick than older adults. Next slide, please. So let's move into the role of discovery. Next slide, please. Okay, so this got me to the point of wondering, okay, well, now we know that within children, there are a bunch of different stuff circulating, and they're probably silently disseminating things. And we don't know who are these children and why, because we said the median age of these children was 4. I mean, these are little kids that haven't even, they may be preschool, but they haven't even started school. They are not in the healthcare system. They are not, that we could tell. So we wanted to really look at that. So what are the exposures and host factors that can predict are predictors of infection within these children? And so we had a 5-center study, so we had the geographic ability to look at this throughout Chicago. And so we divided Chicago up into these regions, as you can see, and this involves the greater Chicago area. So please click. So the first thing I was curious about was these PMFQR, so plasma-mediated fluoroquinolone resistance, because it's so unique from adults and it's usually chromosomal in them. And these are uncommonly used in children. Okay, next. And so I was wondering, because I was interested in these and horizontal gene transfer, are there regional differences, sort of what we saw in MRSA with this? This had not at the time really been looked at in the US, really understanding if there are hotspots for these infections. So we were looking at that, and we looked at CTXM9, which is a different type of CTXM that is usually horizontal gene transfer. It is not a clonal strain, so it's different. And so that's what I was thinking was a problem in children, and we wanted to understand what were the differences here. And next, okay. So yes, so we looked at we wanted to know about community origins and environmental influences for these infections. Next. So these are the papers just that really focuses on this. Next. Okay, so what we found just, we looked and we were wondering if there were going to be regional issues in Chicago area, and we did find that there were different regional issues. What we found was when you looked at regions 5 and 6, children who lived and resided in those regions had an 80 to 90% had 5 times more likely odds of having these infections than those living in region 4, which was our reference region. And those the children living in regions 1 and 2 were at 80 to 90% decreased risk of these infections. And of importance, when we look at these independent risk factors on multivariable analysis, you can see that there was also non-white, non-Black, non-Hispanic race, and this is the time was a checkbox phenomenon. So race other could be mixed race. It could be also races of other origins, which could be important, but this is a separate risk factor, which we'll show you. And also we can see that they were presenting in the outpatient setting. So these were community associated infections. And so this was very important. And importantly, that we did not see any healthcare risk factors that could explain this. There were no differences in comorbidities in the children, medical devices, or antibiotic or healthcare exposures. Next slide. So this meant that the epidemiology of this may differ in children from the traditional adult epidemiology. You can hit next one. So when this really came out in the adult population, this was really thought to be this whole idea of a transfer chain between the hospitals, where there's a chain of transmission between the acute care settings and the long-term care facilities, and these patients who are chronically ill, older adults who are being transferred in between are colonized with these bacteria, and these are moving in between the facilities. And that was the main thought of how some of these drug resistance genes were moving within our communities. You hit the next one, please. So what are the independent associated factors in children, though? They differ. You can start hitting them. So that's fine. You can go ahead. So they presented from the community, and that was different. And then the region of lowest risk, where we saw those infections, which was the region 1, really has the largest pediatric medical center in Chicago. But yet when you looked at that area, it was, the kids were, that is the largest hospital that's in Chicago, and it's where I trained for my residency and fellowship, the largest children's hospital. And what was interesting about this is that the children who were having these infections were being cared for there, but they did not live in that region, although most of the isolates were coming from this healthcare center. And then the region of highest risk, which were where we showed you the other regions, there are no participating centers and really not big children's hospitals. So the race other is that travel to endemic regions, visitors to endemic regions. It could be because we know that international travel is related to that. However, none of the race other cases were residing in the high risk region. So this was an independent risk factor. And again, we saw none of these other exposures that could answer this question. Next slide. So the lesson was for me that at this point, the battle's probably outside the hospital wall. So I had to pivot and really start to look outside of this, where it's a very interesting setting that I'll show you in the next slides to try to have to do research. So the antibiotic reservoirs in the community and beyond are very numerous. And this is what I call the chain of transmission. And you can see here, it's really extensive the way the movement of antibiotic resistance is within the chain. And these are perpetual reservoirs. So they continually are spreading one way to the other and back and forth. Next. So one of the things I wanted to point out is I mentioned those fluoroquinolone resistant mechanisms. One of them was this one called OQA X and B. This is for this drug, olaquindox, which is actually not technically an antibiotic, but it's a growth promoter for pigs, and it's used in pig feed and sometimes in chickens. And so this is one of those plasmid-mediated fluoroquinolone resistance genes, which we'll talk about in a little bit. And this is one of the things we were finding in the children. Next. So this is where One Health comes in. This is very important in understanding community reservoirs of bacteria. So there's an interconnectedness among all of us. And so it's important not to be siloed in our research and in our work outside of one area. So for us, we work with humans, right? But it's important if we want to change the paradigm and we want to understand really what is going on, we need to understand antibiotic resistance in all of these areas, people, animals, and environment. And this is the interconnectedness of all of us. And this is what is called One Health as an approach. And this is where my research has gone. Next slide. Oh, next slide. Okay, so then the next part of this is finding the sources. So we had to go really start with the environment because that was where our hypothesis is this coming from. Next slide. So I don't know if you know about John Snow, but John Snow was a scientist who was in England back in the 1800s. And what he did was look for cholera, and he was trying to understand where it was coming from in the community. And so what we did is he found this Broad Street pump, which was the main source of where people in this one area were getting cholera. And this is one of the first examples of basically geocoding or that kind of research. And so similar to him, we wanted to understand if there were areas or hot spots of infection. And so you can click. So what we did is first started by mapping out the zip codes in the areas where we found the high-risk kids. And what you'll see here is that there are clusters of zip codes that were highest risk. And so what you can't really see very well is in between there is that there was an area that seems to be running in between those, or at least these zip codes seem to be aligning along something. So if you click, what they are aligning around and what you see in that area is actually a bunch of waterways. So what we did is we went to do a pilot study on trying to understand this, looking to understand why it could be that these waterways are related to why we're having these issues with the infections in the children, because these are where the hotspots are. So I'll show you this in a slide, but these are 4 waterways that we ended up looking into. And so one of them is a channel that runs into the edges into the Indiana eventually, but they connect down to the rivers in the Calumet River in Chicago. The Ship and Sanitary Canal is an actual canal that is not meant for humans to be on at all. And that one is runs also into that Calumet area river. And that one is not connected to what you see next to it, which is the Des Plaines River, which also runs parallel to it. And what's important about the differences here is that the Calsag and Des Plaines receive wastewater, but it is disinfected wastewater from the wastewater treatment plants, whereas Ship and Sanitary, because it's not have people on it, has just received sludge. What is important about Des Plaines and Calsag is they are used for recreational water events, but not swimming. They're called limited contact recreation, where people do things like we do here in Atlanta: kayaking, canoeing, swimming. And then number one is a slow, which doesn't receive anything, and it's just next to the CalSAC channel. Okay, next slide. So what we did is go to these waterways, and we physically walked over and got water from them, samples from them, and we did metagenomic sequencing as well as culture-based methods with whole genome sequencing as a pilot to understand what we were finding in there. And so you can click again. Okay, so that is a heat map basically showing antibiotic resistance within these various waterways. And red is just where there's more antibiotic resistance genes. Green is the lower amount, but there's still drug resistance genes, and yellow is somewhere in between. And so we found a lot of drug resistance you can see throughout all of these waterways, including that slow, which doesn't receive any any type of effluent or anything like that, but is next to that CalSAC channel. So A4 and A3, which are Des Plaines and the Ship and Sanitary Canal, they don't have any connections, but what was interesting about them is that they were very similar. So when you look at the total abundances and the microbial communities between those two waterways, they were 97% similar in terms of their microbial communities, even though they're not connected. And there was also similarities between A1 and A2 that were nearby each other. And so when that also correlated when we looked at the Enterobacteriaceae, and you found the highest E. coli numbers were actually in A4, which is ship and sanitary, not surprising because it just gets sludge treated. But it was interesting to see the differences between these levels of bacteria and the amounts of drug resistance we found, and including MCR1 was in there, which is that mobile colistin resistance gene. We found that in 3 of the 4 waterways. Next. And also importantly, we also were able to culture viable bacteria out of these waterways. And so we were able to look at to see what kind of genes we found in these waterways. And so we were able to find plasmid mediated quinolone resistance, as well as these beta lactamase genes that are ESBL producers in the waterways, same ones we were finding in children. Next slide. So if these waterways aren't connected, why would this be occurring? So the Des Plaines and the Ship and Sanitary Canal are really not more than about 100 yards apart. They are not connected, however. So the question would be, why would they have this? You can click. Why would they have 97% similarities of their communities if they're not, connected? So it could be that they were connected through the ground, or there are other sources, which we're about to explore. Next. So this is our beautiful Chattahoochee waterway here, and you can see that there are a lot of people who do different activities on here, and this happens all the time throughout the year, including things like the kayaking and canoeing we talked about. And then it looks like in the bottom right there's some kids who are jumping. I don't know if I'd ever do that one, but certainly people do a lot of activities. Next slide. So this gets into what kind of sources contaminate the waterways. So if you look to the left, what you're seeing there is the wastewater treatment plants are enter into our what we call surface water, so our rivers and lakes. That's where this discharge comes out into the water, and it's supposed to be disinfected. But there are some ARGs that, as you could see from my prior graph or work, that they do actually enter into the water. The question is, how do they actually impact us as humans, right? So you can see that there's industrial water, wastewater. You look to the right, you see these cows. So this is agricultural. So the cows are in their little stream. That stream runs also into these surface waters. There's a nice dog. And then waterfowl actually are also someone who can carry around these genes, and they fly obviously in between these waterways. Okay, next. So this gets to what we call point source and non-point source discharges into the water. So point source is what you see to the left. That is where, there's literally dumping straight into these waterways that we use. And then there's the non-point source. So that's runoff that comes from these farms and streams where you're putting manure onto the farm, and then it rains, or you're irrigating, and that comes into our streams, and that comes out. And then the animals also. Next. So the other thing is a question about not just surface waters, which are what you see to the left, but groundwater. So groundwater is also something we use for our drinking water. And so that actually is that is something people have been using more and more. And now that's starting to become a question about its actual contamination as well. It never thought to be before, but now it's starting more and more to think the groundwater is also getting contaminated. Next. And then this is another thing that we deal with, right? Flooding. So flooding coming into your households and things like that. Next. So this is just showing something that I think is important here in Georgia in Atlanta area and in Chicago. We have what are called combined sewer and storm drain systems. Those are where you have when it rains, there is sewage and stormwater that come out all together into the through the pipes into the creeks or waterways. That's different from separate from when it rains and you have a separate sewer where it's just stormwater that comes out through the downspout separate into the waterways, and then the sewage actually stays underground and there's treatment that way within the sewer system. Those are, as you might expect, more expensive, and so many places have these combined combined storm drain and sewer systems. And the problem with that is if that gets backed up, what happens? We get flooding on our streets, things like that happen. So these are where contamination events can occur. And to the right, I'm just showing you a couple ones that have been recent. The one was last year was the E. coli contamination over the holiday of July 4th. And then this was just last week where they talked about the Chattahoochee having high E. coli levels. And this was actually some breakdown in one of the wastewater treatment plants where there are cracks and things, and there was sewage coming into the Chattahoochee. Next. So let's move on to the importance of community and households next. So we want to learn from our colleagues. This is a dear friend of mine, Stephanie Fritz. I'm working on a project with her funded by the NIH. And she started out doing work on trying to understand about transmission of some of these community-acquired pathogens in the household with MRSA, which is the first one where we really acknowledge the role of the community in this, but we didn't understand its transmission dynamics. So Stephanie did a couple studies trying to understand the movement between index patients, which were children, and their household context and pets, and the role of environmental surfaces in movement of bacteria. Next slide, please. So these are just a couple of her papers that were very important in this next slide. Okay, so when you looked at movement of bacteria, what she did is she wanted to see surfaces. She looked at movement between parents, movement between environment and parents, movement between parents and their children. And you can see that the movement of bacteria and transmission of bacteria within a household is complex, and that it happens between people and between the environment and people. Next. So when you look at this, you can see that really household surfaces really were the sole source of transmission in about 35% of events that happened. So that's where it was coming from. And I think that was really something that was very enlightening, that the surfaces really could be part of the problem in our houses to transmission of us getting the infection. But clearly we transmit also to each other. And that there was, when you had a higher burden of contamination on your surfaces, you were fourfold more likely to have transmission that was occurring. So this was the first step to understand about MDR transmission. But if you click, this really told us that there was a problem with MRSA in households, right? Something we know is on our skin. But next. The question was, is this a problem with the drug, with just gram-negative bacteria? And that was something that was unknown. Next. So when you look at this, I'm sorry, just go ahead and click through. Let me see. Next. Okay, sorry, no, next one there. So you can see, sorry, could you go back? There, I'm sorry. So you can look and see that what they did is they looked at different transmission sources, of transmission, and you can see they looked at various things, and there were differences between which surfaces had more potential for transmission than others. But there were several surfaces that had multiple ways of transmission. Next. So we started this study, as I was talking about, it's funded through NIAID, looking at whether or not our households had ESBL in them. So really understanding the prevalence in molecular ecology. And we just really started this study in the last few months. So I'm going to show you some of the data, early data from this study. And really, the next step will be after we look at which surfaces are involved in which bacteria we're finding what are the individual level risk factors for infection in children. Next. So this is my couple of people from the lab I just want to recognize for all the work done here. The middle just shows you the surfaces that were actually looked at. So it's a bunch of different surfaces, from the kitchen to the bathroom to the living room, to people's handheld devices. Next slide. And bed sheets also. Okay, so this was data from a couple weeks ago. Where we are. So far we have looked at about 120 households. And what we did is, because it was a MRSA study, there was only inguinal fold. So when you want to look for colonization in humans with Enterobacterales, the best way to do that is perirectal or rectal or stool samples. However, in this study, this was a skin bacteria that was being looked at MRSA. And they had inguinal fold samples, and that's not an unreasonable proxy because oftentimes the groin is colonized, but it does decrease your sensitivity of finding those organisms. And then we've looked at over 2,000 household surfaces at this point. And so the first graph really just shows that most every surface in the household has and person has bacteria on them. And that there's a lot of gram-negative bacteria on surfaces in people. The Enterobacterales is that purple line, so there's less of that. And you can see to the right the actual percentages of gram-negative bacteria, and I'll break down the Enterobacterales in a second, and the resistant. But you can see that most places have gram-negatives. Next slide. And then you look at Enterobacterales and you start to see there's certain surfaces that stick out that have these Enterobacterales. You can see that the kitchen sink faucets seem to be a problem, sofas, and also things like bed sheets were really important. The oven door handle, some of these were actually different than what they were finding where the MRSA was sitting. And so when you looked at the antibiotic resistant Enterobacterales, you can see a similar type of trend. The people were colonized up to about 3% of people having these drug-resistant organisms, but they were less, maybe 2% or so when it comes to our ones of interest that had the phenotype of cephalosporin or a broad-spectrum cephalosporin resistance. Next slide. So when you look at the bacteria, what was interesting when we were looking for mostly E. coli, right? But what we were finding was a lot of Enterobacter, which was really interesting when we came to look for drug-resistant bacteria. And so really, this is just showing that there were diversity in the bacteria that we were finding in general within gram-negatives on the left. But when you looked at the percent of Enterobacterales that were drug-resistant, you really found that Enterobacter stood out. Next. So here again, just looking at some of these surfaces and what are the most common ones that are drug resistant. Again, you can see this Enterobacter, which I'm going to tell you why is a surprise in a little bit, but you can pass this next one. It's okay. It's not important. But what I want to show you is what's interesting about Enterobacters are the mechanism of beta-lactam resistance in them should be chromosomal. It's called an AMC cephalosporinase. It's not usually an ESBL producing organism. That it can actually pick up these genes, but it was surprising to see this much in the household because many of us think of this as more of a bacteria that we see in healthcare settings rather than something that would be transmitting in households. So we did some sequencing on some of the earlier isolates, and this is Dr. Green and Babaker who helped me with these results. Next slide. So I won't go into this, but this is our sequencing genomic analysis pipeline, and we use SNP related matrices to understand clusters of infections. Next slide. You can skip this. So just to know, single nucleotide polymorphisms really is a mark of genetic resistance. So if it's lower, the amount of SNPs or variants between these really if the lower the count, that's more likely how similar the isolates are. So for us, a cluster or transmission definition is less than 15 SNP difference between two isolates. All right, next. So this is showing a phylogenetic tree of relatedness of between 4 households of bacteria that are just Enterobacter. You can see the surfaces over to the left, and it just shows you that there's a diversity in the surfaces and the and people that we were seeing these Enterobacter on. And so what's interesting is we found 3 different clusters among these households, and they were happening among different surfaces. One thing that was interesting is we found one instance where there was apparently a locally circulating Enterobacter because they look very similar in relationships, and that's the 527 and 522. That's kind of about 7 lines down. So there's some circulating strain there of Enterobacter within their community, and we have to look and see if these people are related. But what's important is the bed sheets are very important in this transmission of bacteria, and we're finding a lot of that. So that might be one of the roles between people and the environment where we start to see these transmission routes. And we know that these are moving between people and their surfaces. Next slide. So what thing was interesting is that we were finding these plasmids within these Enterobacter. And again, like I was saying, these, a lot of times people think of Enterobacter as having chromosomal mechanisms of resistance, but clearly these are conjugative plasmids within these Enterobacter that are participating in this. Next. And so that OQX, that olaquindox, we also found that within the Enterobacter within these households, along with other AMR and metal resistance genes. Next. So this is a St. Louis study. So this means now we found this in children in both in their households now in both St. Louis and Chicago communities. Next slide. So you can click. The reservoirs of Enterobacter are numerous. And so basically the interesting thing about Enterobacter is while we think of it as a bug that we see in healthcare settings in people who have been on antibiotics, it actually turns out that these are actually in the environment and in households and in humans and all over the place. They're ubiquitous, and I think we don't think a lot about these. You can click. And there are a lot of, so the point of this is that I think that there are household issues of transmission that are actually impacting that. So I've added that. And then just knowing that we're finding these other plasmid-mediated fluoroquinolone resistance genes within other areas, not just Chicago. And we know that these co-spread with other resistance genes. So this is an important finding, I think, going forward for us. Next. Okay, next. Okay, so just to point out that again, we found that these are circulating among kids. The community acquisition is important. There are known healthcare exposures. There are high and low risk regions for these infections, and the reservoirs are really unknown. Next. So when we look at our chain, you can go ahead and click. You can see that the household probably is important in the chain of transmission, and that there's exposures that are occurring in the environment and with animal reservoirs, and ongoing transmission is probably being facilitated through spreads in households in the community. And so these are the areas of my research and going forward where we're looking at environmental sources, community spread, and also geospatial links to infection. So this is why One Health is important. Next. So you can skip, but this is basically just collaboration is critical. And I just want to thank all of my collaborators. So you can go ahead and click. Okay, so we define the problem. So we looked at the importance of surveillance and linking organisms to disease. And we looked at the understanding of the organism, the importance of studying mechanisms of resistance. Important is the role of discovery. We have to link these genomics of the bacteria to the clinical epidemiology. Then we went to the sources. We went to the environment to try to understand the community and the spread of resistance. We were trying to understand health implications, and that's important of all of your research, our work, and in identifying risk factors and implications of MDROs. And again, collaboration is very critical, and it's really important not to be in a silo, and that this has to be a One Health approach and requires teamwork. So next. Just a thank you to everyone who's worked with me over the years. And I'm sure I probably forgot someone, but I really appreciate your time, and I'm very apologetic for all of the issues that we had. Thank you.